So if we look on the schedule, we are working on chapter 5 today, microbial metabolism. This week in the lab, finish lab 6. Hopefully you already finished it, but we started it on Tuesday. And then turn in the worksheet for lab 6 by Saturday. And we did discuss and started the unknown project. I did email everybody your unknown bacteria, and I think I emailed the instructions, not the, just a few instructions, not the document that is the instructions for the project. Any questions about anything? Did anyone not get your unknown bacteria number? Okay, so get started on the unknown project. You have until nearly the end of the term to work on it, and you're waiting before you get the first results. I'll be sending those on Tuesday. But you can start thinking about the project. I guess you can't really get started on the project other than you should put your unknown number on the unknown notes page, because if it's not there and you ask for test results, I'll word you back that I can't give it because I need your unknown number and you don't have it on your unknown notes. All right, let's close this down. So we were talking about enzymes, and we mentioned last time that enzymes normally end with ACE. This slide is showing you that enzymes sometimes do not work alone, that sometimes they have a coenzyme or a cofactor that get together with the enzyme to make the complete enzyme. And you do not need to know the name, like the apoenzyme and the holoenzyme. All you need to know is that some enzymes take a coenzyme or a cofactor to become complete. A coenzyme is a biological molecule, like a protein that adds to the enzyme. There are a few common coenzymes which I'll show you in the next slide. One of them is NAD+. That gets together with the enzyme to make it complete. And a cofactor is usually an ion of a metal, like iron or zinc or magnesium. Even calcium is a metal. So cofactors are simpler and smaller. A coenzyme is a biological molecule. Some important coenzymes are shown on this slide. You only need to know the first two because we're going to talk about them when we're talking about aerobic respiration, which will be later in this lesson. I'm not sure we'll get to them today, but we will get to them. NAD plus, or nicotinamide adenine dinucleotide, and FAD, flavine adenine dinucleotide. We'll talk about them with aerobic respiration. Those are important coenzymes that we use in aerobic respiration. Some other important enzymes are NADP plus, which you don't need to know, FMN, and coenzyme A. And to tell you the truth, we do talk a little bit about coenzyme A, but I'm not going to ask you to remember that one as an important coenzyme. So the enzymes, how they work is sort of like a lock and key, and that is, I don't know if the enzyme is the lock or the key. And that is, it gets together with one substrate, and the substrate fits the enzyme, or you could say the enzyme fits the substrate, like a key fits a lock, meaning one key fits one lock and opens that lock. One substrate fits an enzyme, and the enzyme can convert that one substrate into the products. And they do bind together, meaning the enzyme actually binds to the substrate to make a complex. And that puts stresses on the bonds of the substrate, converting the substrate into the product. The product then falls off of the enzyme, and then the enzyme is then free to bind to other substrates. And this cycle can happen thousands, tens of thousands, hundreds of thousands, even 1,000,000 times. Okay, any questions about that? You don't need to know the turnover number. It's usually a large number, like 1 to 10,000 substrates per second. This picture, I'll blow that up a little bit, is showing you that an enzyme isn't really as simple as a lock and key model, that it's really closer to a hand and glove model, and that is the enzyme is the glove that goes over the substrate, sort of like a glove goes over your hand, and then the glove molds to the shape of your hand. And you can sort of see that here, that the enzyme changes shape when it binds to its substrate. What else can you see in this picture besides that the enzyme changes its shape when it binds to the substrate? Okay, come on, what else can you see in this picture? There are two things, although one of them is you have to be a really good observer to catch. Hello? What two things can you see in this picture?
Does it get smaller?
Say again?
Does it get smaller?
You're talking about the substrate
Yeah.
or the enzyme. What are you talking about?
Good question. Maybe the enzyme?
The
Well,
enzyme
the enzyme is changing
shape
its
is changing.
So yes, in this case, it's getting smaller because it's changing its shape. But it's not really smaller. It has the same atoms in it. Those don't change. Well, something that's very obvious is the enzyme is many times the size of the substrate. I'm surprised nobody noticed that. That's quite apparent in this picture. And that is true. The enzyme is usually much bigger than the substrate. Take a look at the configuration or shape of the substrate here and there. Does anybody notice a difference? Well, I'm really disappointed in you guys. Nobody is stating anything. The substrate does change its shape slightly. If you notice, it's a little flatter here than there. And it is true, you kind of have to blow that up to see that. That's as big as I can get it. I don't know if that made it bigger for your end or not. And so the important part is that, oops, I went too far. There we go. The important part is that even the hand and glove model is a little oversimplified in the way that enzymes and substrates work. A better way of saying it is the enzyme fits the substrate sort of like the glove fits the shapeshifter's hand. On Star Trek Deep Space Nine, there's a shapeshifter, and he can change his shape, and that is the glove is molding to the shape of the hand, but the shapeshifter's hand is molding to fit the glove, changing to fit the glove. Is that clear? Those of you who watch Deep Space Nine will understand my connection there. Any questions about any of that? Hopefully you saw that the substrate is changing to fit the enzyme, and the enzyme is changing to fit the substrate in the enzyme-substrate complex. And that complex is putting stresses on the bonds or bonds of the substrate, and that's what changes the substrate into the product. In this case, we're seeing that the substrate is breaking into two to become two products, but it could be that two substrates are going to join to form one product. All right, any questions about any of that? This slide is very important, so make sure you understand it. All right, so this is the beginning of the start of worksheet 5, so I'm going to go over only the main portions, and I'm going to go over the main portions because some of you aren't doing very well on your lecture worksheets, so I will briefly discuss them. There are various factors that influence an enzyme's activity, and these factors will change how well the enzyme works, or could completely denature the enzyme, in which case the enzyme isn't going to work at all. And these are listing the different factors that can do that. We'll also talk about inhibition of an enzyme, which can totally shut down the enzyme, and then feedback inhibition. So what happens when an enzyme becomes denatured is it changes its folding pattern, and if we say it's 100% denatured, the enzyme isn't going to work, and it'll be a totally different folding pattern of a protein as shown here, and this is the amino acid sequence, and it's definitely not folded properly. Different proteins can denature if the protein is at the incorrect temperature, the incorrect pH, or the incorrect saline concentration. And the further off it is, meaning the further the temperature is from the optimal temperature, the more the protein will denature. This is showing you that generally speaking, enzymes have an optimal temperature or an optimal pH, and when you move away from that optimum, the enzyme still functions, but as you move further and further away, the enzyme will function less and less until eventually the enzyme will stop functioning. Any question about that? Substrate concentration can also affect how well the enzyme works. The higher you have the substrate concentration, the more likely it is going to collide with the enzyme, and then you'll get an enzymatic reaction. And when the substrate concentration is very low, the enzyme isn't very active, And as you increase the substrate concentration, the enzyme activity will increase up to a point. Why, at this point, when we increase the substrate concentration, why do you think that the enzyme activity has not increased at all?
Does it have to do with limited enzymes?
Yeah, the enzyme is limited, and if all the enzyme is bound to the substrate, you can't increase the enzyme activity by adding further substrate because all the enzymes are already bound to a substrate. Is that clear to everybody?
Yeah.
Okay, thank you for answering. I'm moving on. Competitive inhibition is when there's a drug or something that competes for the active site with the substrate. So the two, the substrate and the competitive inhibitor, are binding to the same site in the enzyme. And that's why it's called competitive. It's competitive inhibition. Okay? Some drugs act that way, where they bind to the active site of the enzyme. Usually the drugs don't bind, what's the word I'm trying to say, forever, that they'll fall off, and so you need to keep taking the drug and keep it in your system. There are some poisons, though, that the competitive inhibitor totally binds to the active site and it totally inactivates that enzyme. And of course, those poisons or toxins are very toxic to us because they shut down our enzymes. You can survive that. What happens is your body can make more enzyme, assuming that there isn't more of that poison. There's also non-competitive inhibition. It's also called allosteric inhibition. So these two words, non-competitive inhibition and allosteric inhibition, mean the same thing. That's when the inhibitor binds at another site of the enzyme that is different from the active site. How that works is when the non-competitive inhibitor binds to the enzyme, it changes the folding pattern of the enzyme. So now the active site can no longer fit the substrate. And then lastly, feedback inhibition is a type of non-competitive or allosteric inhibition. And what happens is usually the end product that's made in a chemical reaction, which may be a metabolic pathway that makes the end product. In this case, we have enzyme 1, enzyme 2, and enzyme 3. And the enzyme 1 and enzyme 2 make intermediates. Enzyme 3 makes the end product. When enough of the end product is made, it will then bind usually to the first enzyme, changing the folding pattern of that enzyme, so the enzyme's active site could no longer bind to the substrate. And that will shut down the enzymatic pathway, because if you don't make any intermediate A, you're not going to make intermediate B, and you will not make the end product. Feedback inhibition is frequently seen in cells because it's the most efficient way to inactivate a pathway. You shut down the first enzyme, and it's the end product that shuts it down. Then none of the intermediates are made. If you were to shut down the second enzyme, you would make intermediate A, and that would be costing the cell energy. Okay? Does everybody understand feedback inhibition? Anybody not understand it?
Okay, thank you.
All right, it's frequently seen in cells because once we make enough end product, we don't want to make more of the end product. And the end product itself will shut down usually the first enzyme. When the cell needs more end product, how is more end product made? Anybody have a guess? Somebody was going to say something.
Could you repeat the question, please?
If the cell needs more end product and yet it shut down all of the enzyme number 1, how does the cell make more end product? It's actually shown in this reaction, but you got to put a little bit of common sense to it.
I think it looks like by the process of isozygosis.
I'm not sure what that means. Could you explain the process of zygosis?
Sure. I learned that in my last quarter in biology.
That could be. I'm not sure of that term, so could you educate me?
Basically, the cell will incorporate, a new molecule, and kind of multiply by that process. I think so. Yeah.
Okay, I didn't hear that very well, so I'm not sure if that is the correct answer or not. What happens is the end product binding to the enzyme is not irreversible. Every once in a while, the end product will fall off. And while there's lots of end product around, another end product will bind to the enzyme and keep it inactivated. But if there isn't other end product around, it won't bind to the enzyme that the end product has fallen off of that enzyme. And then that enzyme will become activated and will start making the products necessarily to get more end product. And with more and more time, more of the end products will fall off of the enzyme 1, and there won't be enough end product around to inactivate those enzymes. And so more end product will get made. Is that clear to everybody?
Yes.
All right, thank you. This one I need to talk about. This is actually kind of important. And that is there are enzymes which are not protein enzymes. Most enzymes, probably 95% of enzymes are a protein. But some enzymes are RNA enzymes, and that's what I call them. It's an enzyme that is an RNA. It's not a protein. There's another name for that. They're called ribozymes. And what a ribozyme is an RNA enzyme. It's an RNA that cuts and splices RNA or another molecule. They function as enzymes in that the RNA actually has an active site that binds to the substrate, and the enzyme is not used up or modified in the chemical reaction. So once that RNA enzyme has made the product, that RNA enzyme is then free to bind to another substrate and work as an enzyme again. Okay, any questions about that? There is actually one RNA enzyme that we're going to talk about. Tutora, your textbook author, does not mention it, so I'm going to mention it. And that is, we do talk about I'm drawing a blank here. Ribosomes. There we go. Ribosomes make proteins. Well, there's an activity where the amino acid is removed from the tRNA and then linked to the growing amino acid polypeptide to add that amino acid. And that action of the ribosome is by an RNA enzyme. And we'll talk about that a little bit more later in the term. I don't think we've talked about ribosomes and how they make a protein, how they add on amino acids. Is that correct? Anyways, the ribosome, in its activity with the amino acids and moving them from one location and binding them to the growing polypeptide, that is done by an RNA enzyme. And that's the only RNA enzyme we will discuss in this class. But there are some enzymes which are RNA, and that's what you should know here. They're not all always protein. All right, any question about any of this? Because if not, we're moving on to another topic in Chapter 5, Metabolic Diversity Among Organisms. Any questions? Last chance. All right. So we can classify all organisms by where they get their energy source and their carbon source. If an organism, and we're talking mostly about microorganisms, get their energy needs, well, I guess I don't have that shown here. If they get their energy needs from light or the sun, we call them a phototroph. They derive their energy from light. And green plants, I think everybody knows, are phototrophs. And then if they don't get their energy source from light, they get it from a chemical, we call them chemotrophs, deriving their energy needs from an inorganic or an organic chemical compound. And we are a chemotroph. We get our energy needs from the energy in the organic molecules in our food, such as glucose. Any questions about that? You can also classify organisms basing it on where they get their carbon source. Organisms are classified as autotrophs if their principal carbon source is from CO2 in the air or dissolved in the water. Like a bacteria in the ocean is going to get its CO2 from being dissolved in the water. And everybody knows green plants are, at least most of the time, autotrophs, getting their carbon from carbon dioxide in the air. We, on the other hand, cannot do that. We are a heterotroph. We get our carbon source from an organic chemical molecule such as glucose. Any question about that? Now, if you're ever confused in the more complicated scenarios, always come back to finding out where does the organism get its energy source, because there's only a phototroph or a chemotroph, and where do they get their carbon source, because there's only autotrophs or heterotrophs. So if you're ever confused, just go back to this, because these are the simplest ways of classifying all living organisms. including the microorganisms. But we can combine the terms and where organisms get their energy sources and their carbon sources, in which case it's a little more complicated. If you classify on both the energy and the carbon source, you have the following 4 classifications: photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs. And there are differences in the photoautotrophs. There are oxygenic photoautotrophs and anoxygenic. And then there are differences in the chemoheterotrophs, and it's not shown, there are differences in the chemoautotrophs too, and that is the chemical compound giving carbon can be an organic chemical molecule, which would be like glucose, or it can be an inorganic chemical molecule, such as it could be a nitrogenous compound, an iron compound, sulfate or sulfur compound, for where they get their energy needs. So looking at this again, I think everybody knows green plants are photoautotrophs. They get their energy needs from light, and they get their carbon from carbon dioxide. But please understand there are some bacteria as well as algae which are photoautotrophs. Any question about that? Cyanobacteria is a photoautotroph which makes oxygen, so we call it oxygenic photoautotroph. But there are anoxygenic photoautotrophs. I won't ask you about them. The green and purple sulfur bacteria they're photoautotrophs, but they do not make oxygen. All right, the next easiest one to talk about, because everybody knows about them, are chemoheterotrophs. And you know about this one because you are a chemoheterotroph. You get your energy needs from a chemical compound, such as glucose. Although there are other chemoheterotrophs which get their energy source from an inorganic chemical compound. Like a chemosynthetic bacteria can get their energy needs from a sulfur compound like sulfate, or a nitrogenous compound like nitrate, or iron, what do you call that? An electronic configuration of iron. It just changes the electronic configuration of iron so that there's fewer electrons. All of the chemoheterotrophs get their carbon source from an organic chemical compound, such as glucose. And I mentioned that the animals are that way because we are. Protozoa are that way. Fungi are that way. Some of the bacteria, like E. coli, is that way. The fermentative bacteria, the ones that make yogurt or cheese, are that way. Okay. Another one, as I mentioned before, is some of the chemosynthetic bacteria, which gets its carbon source from glucose, but gets its energy source from an inorganic chemical compound. So some of the chemosynthetic bacteria are chemoheterotrophs. And some of the chemosynthetic bacteria are chemoautotrophs. So the chemosynthetic bacteria are here and here. Most of them are chemoautotrophs. Chemoautotrophs get their energy needs from a chemical compound, an inorganic chemical compound like H2S or sulfate or a nitrogenous compound or iron. We have the iron oxidizing bacteria listed down here. they get their carbon source from CO2. That's why they are an autotroph. These are the chemoautotrophs. And some, probably most of the chemosynthetic bacteria are chemoautotrophs. Okay? Any question about that? If you never heard of the iron oxidizing bacteria, you've actually seen them. Take a look at your toilet, and if you take off the tank, the water tank's lid, and look inside, you'll see this yellow or brown stuff on the side there. That's iron oxidizing bacteria or some other chemosynthetic bacteria, because that region gets no light, so they're not photoautotrophs. There's no glucose or organic chemical compound in that water. So that bacteria and other sludge there is largely iron oxidizing bacteria, utilizing iron in the water to get its chemical energy needs, and then its carbon needs comes from carbon dioxide dissolved in the water. And I know you've all seen those chemoautotrophs before. Okay? Any questions about that? Now, the photoheterotrophs are the most complex because none of you have ever come across a photoheterotroph unless you happen to study it maybe in Biology 160, because these are not common organisms. They get their energy source from light, that's why they're a phototroph, and they get their carbon source from an organic chemical compound such as glucose. Some photoheterotrophs are the green and purple non-sulfur bacteria. And have any of you ever heard of the green and purple non-sulfur bacteria?
I don't know if I have.
No, probably not. Anyways, they're photoheterotrophs, and I won't require you to remember that the green and purple non-sulfur bacteria were photoheterotrophs. You should just know that a photoheterotroph gets its energy needs from light and its carbon needs from an organic chemical compound such as glucose. All right? Any questions about that? No questions? Then let me state that for most chemoheterotrophs, both the energy source and the carbon source are usually the same organic chemical compound, such as glucose. And for us, and for animals, that's always the case. But for the chemosynthetic bacteria, it's not the case. But that's only a few of the chemoheterotrophs. For most of the chemoheterotrophs, they get both their energy needs and their carbon needs from the same molecule. Okay? Any question about any of that? Now, you need to know the nutritional classification of organisms, but you only need to know that what's outlined in red here. So all organisms, let's blow this up. All organisms can be classified as all organisms, and we can classify them in their energy source. There are phototrophs if they get their energy needs from light. There are chemotrophs if they get their energy needs from a chemical compound. If they get a chemotroph, if they get their carbon source from an organic chemical compounds, we call them a chemoheterotroph. If, on the other hand, they get their carbon source from CO2, we call them a chemoautotroph. And you can further break down the chemoheterotrophs by where the final electron acceptor is coming from and whether they're oxygenic or not. We're not going to I'm not gonna test you on that. All right. And the chemoautotrophs can be broken down by where they get their hydrogen source. We won't look at that. For the phototrophs, besides their energy needs from light, you can break down their carbon source. If it comes from an organic chemical compound, it's a photoheterotroph. If they get their carbon needs from CO2, they're a photoautotroph. And then you can break the photoautotrophs, and that is, are they oxygenic or are they anoxygenic, meaning they do not generate oxygen, and they're a photoautotroph. Any question about that? All right, if there's no questions, let me warn you that this is a little bit of an Obler simplification. There are complications, and that is an organism can change its classification depending on what is available. Some of you may have studied Euglena. It's this green little protist that is found in pond water. And in the pond water, if there is no sugar dissolved in the water or no organic molecule, we'll just say sugar, then as long as there's light, the euglena will act as a photoautotroph, sorry. And that is, it'll get its energy needs from light and its carbon needs from CO2. So euglena is perfectly happy working as a photoautotroph. But on the other hand, if you take away the light and you add sugar to the water, euglena will switch acting as a photoautotroph, because it can't act as a photoautotroph, there's no light. It'll start acting as a chemoheterotroph, because it can get its energy needs from glucose and its carbon needs from glucose. Okay? And then again, if you add the light back and there's glucose in the water, it can act as a photoheterotroph, getting its energy needs from light and its carbon source from glucose. Any question about any of that? An organism can change their classification depending on what they're doing and what is available. And I gave you a good example, euglena, which can change what it does. Okay? There's also the mixotrophs. Many of the mixotrophs are the chemosynthetic bacteria, and the mixotrophs are bacteria or archaeobacteria which will use whatever source is available. And usually they will use whatever source is available at both times. Like, for example, if glucose is available and CO2 is available, they will use both carbon from CO2 and carbon from glucose. So they're acting as a, let's see, a photoheterotroph or a chemoautotroph or a chemoheterotroph. All right? And they can also use, if they are a mixotroph and they're photosynthetic, they can also be a photoautotroph or a photoheterotroph or a chemoautotroph or a chemoheterotroph. And sometimes they'll do all of these four at the same time, meaning they'll use whatever is available to them. And sometimes they will do it at the same time. Any question about that? Like I said, the chemosynthetic bacteria, which are chemoheterotrophs or chemoautotrophs, they will frequently do that. They're mixotrophs, and they will use both glucose for a carbon source and CO2 for a carbon source. Any question about that? That is a complication that you just need to know about. That organisms can change their classification? Can humans change their classification? I'm
No.
asking a question. Can humans ever be a photoautotroph?
No.
Thank you. Yes, humans cannot change our classification. Bacteria are a little different. All right, so that's the classification of living things. You should know that energy is associated with the electrons that form the chemical bonds. And so all nutrient molecules have energy in their chemical bonds. And that is how we get our energy needs from glucose. We break the chemical energy bonds in the molecule of glucose, breaking each carbon bond, by the way, and we harvest that energy from glucose. Any questions there?
No.
Cells need energy, and it's obtained from catabolic reactions, and that energy is usually stored in the bonds of ATP. And then ATP is the energy carrier of cells. Usually when a cell needs energy, it'll get its energy needs from the molecule of ATP. ATP has high energy phosphate bonds, which high energy means they're unstable, and when that high energy phosphate bond is broken, it releases a large amount of energy which the cell uses for its energy needs. And that energy is released quickly and easily. That's why ATP is used as the energy carrier in cells. Any questions about any of this slide?
No.
Thank you. All right, so we get our energy needs from oxidation reduction reactions, but in reality, almost all chemical reactions, probably all of them, but we're going to just say almost all chemical reactions happen by oxidation reduction reactions. Oxidation is when one molecule or atom loses an electron. So oxidation is the loss of electrons. Reduction is where the other molecule or atom gains an electron. Oxidation reduction reactions are always coupled, meaning one molecule or atom loses an electron and another molecule or atom gains the electron. And for some reason, they decided that oxidation reduction reactions was just too long to say, so they shortened it to redox reactions, and re from reduction, ox from oxidation. I guess that's red from reduction. And it just is the reverse. Reduction goes before oxidation when we say redox. So redox means oxidation reduction reactions. One molecule loses the electron, so it's oxidized molecule A here. B gains the electron, so it becomes reduced. A is oxidized, B is reduced. An example, which is showing you in atoms, copper plus 1 in the reaction with iron plus 3, the copper becomes oxidized, loses an electron, so it's changed from copper plus 1 to copper plus 2, and then the iron becomes reduced, gains an electron, and it becomes iron plus 3 to iron plus 2. Okay? Any questions about that? Now, this is a mnemonic device for helping you remember oxidation reduction reactions. You can say, Leo the lion goes grr. Leo, lose electrons, oxidation. The lion goes grr, grr, gain electrons, reduction. Leo the lion goes grr. Any questions about that? Now, oxidation reduction reactions in biological systems, usually the proteins, an H plus ion, are usually removed with the electrons. Remember, an H plus atom, if you remove the electron, all you have left is a proton, the H plus ion. Any question about that? So biological oxidations are often called dehydrogenations because when you lose an electron from this starting molecule and it's gained by this molecule, NAD plus, the coenzyme, the NAD plus becomes reduced, gaining the electron, and then the other molecule becomes oxidized, losing the electron. But usually the hydrogen ion follows the electron and will be gained by NAD plus, becoming NADH, gaining both an electron and a hydrogen ion. And usually in biological systems, the H plus ion follows the electron. Okay, any question about that? So, biological redox reactions are used in catabolism to extract energy from nutrient molecules. So when we break down glucose in biological redox reactions, meaning oxidation reduction reactions, we obtain the energy in glucose for our energy needs. Highly reduced compounds, meaning compounds that contain many hydrogen atoms, are typically degraded, catabolized, to become highly oxidized compounds. So glucose, C6H12O6, and if you don't know that is glucose, you should learn it. Glucose is C6H12O6. Is a highly reduced compound. It has many hydrogen ions. And it will become oxidized into CO2 and water, molecules that have much less hydrogen ions in them. And then the resulting energy is largely trapped by ATP. And realize when I say largely trapped, the majority of that energy is actually lost as heat, but we do a fairly good utilization of converting that energy into ATP. And we'll talk about the efficiency of that conversion of the energy into ATP. Highly reduced compounds, like glucose, contain a large amount of potential energy that is stored in the chemical bonds of the molecule. Okay? Any question about that? And we can use that potential energy by burning glucose. No questions, I'm moving further. All right, so we're going to start talking about aerobic respiration. I don't think we'll finish this. We'll see. Aerobic respiration is the energy conversion in not all cells, but many cells. If the organism can use aerobic respiration, it will take glucose and oxygen and convert the energy in glucose into ATP using aerobic respiration, which is also called cellular respiration. And I'm afraid you have to know both of those terms, cellular respiration and aerobic respiration. Converting the glucose and the oxygen into carbon dioxide and water. The summary reaction for aerobic respiration is glucose, C6H12O6, plus 6 oxygen molecules and 38 ADP, adenosine diphosphate, and 38 phosphate, the I stands for inorganic phosphate, can be converted into 6 carbon dioxide molecules, 6 CO2, and 6 water molecules, and 38 ATP molecules. This is the overall summary reaction for aerobic respiration. We say it's summary because there's a large number of reactions that occur in aerobic respiration. And not only are there chemical reactions occurring in aerobic respiration, but there are also physical reactions occurring. Any questions about that? You should know this reaction. So when we make ATP in making our energy production, some of the energy from redox reactions is used by the cell to make ATP. ATP is generated by the phosphorylation of ADP. Phosphorylation is just a term that means the addition of inorganic phosphate, and here we have inorganic phosphate, BO4 and 3 negative charge. ADP, adenosine diphosphate, plus energy, plus inorganic phosphate will be converted into ATP, adenosine triphosphate, having 3 phosphate. This is a reaction that takes energy. You need energy to add that 3rd phosphate onto ADP. In animal cells, this reaction mainly occurs in aerobic respiration. It can also happen, at least in your muscle cells, by fermentation. And we'll talk about both of those, not today, but later in the lesson. The squiggly line represents a high energy bond. And if the cell wants energy, it can break this 3rd phosphate off, generating energy. That's why ATP is the cellular currency in the cell. And if the cell wants even more energy, it can break off the 2nd phosphate. Both of those are high energy bonds. All right, any questions? If not, all cells have 3 mechanisms for making ATP. The first mechanism that a cell can make ATP is by substrate level phosphorylation. You transfer a high energy phosphate from a phosphorylated compound directly to ADP. This does happen in fermentation and glycolysis, substrate level phosphorylation. So here we have a 3 carbon molecule with a high energy phosphate, and this phosphate comes off and joins ADP. So ADP becomes phosphorylated, and then we have a 3 carbon molecule and ATP, substrate level phosphorylation. Any questions about that? The second way that cells can generate ATP is by respiration. It's also called oxidative phosphorylation. It involves the sequential transfer of electrons from an electron donor, like glucose, to the final electron acceptor, such as oxygen, and the electrons are donated by glucose to other molecules in aerobic respiration, and eventually end up as the final electron acceptor, oxygen. So the electrons flow from glucose to other molecules and then end up at oxygen, which the oxygen will take the electrons and the hydrogen ions to be converted into water. And those electrons, it's not showing, it starts from glucose, and the electrons flow to NADH, and some of them flow to FADH2. We'll talk about that. And they flow to this molecule, and then to that molecule, and then to this molecule, then to that molecule, then to this molecule, then to that molecule, then to that molecule, and then they end up at oxygen. You don't need to know any of these molecules except for oxygen. Well, I suppose you should know water too at the end, and NADH, and we'll talk about FADH2 later, and I suppose you should know glucose. Okay, and we'll show you aerobic respiration starts with glucose, and the electrons will flow to oxygen to become water. And we'll talk about NADH and FADH2. We won't really talk about any of these molecules. The sequential transfer of electrons happens with the electron carrier molecules in the electron transport chain of aerobic respiration, and we will talk about the electron transport chain later in this lesson. I don't think we'll get to it today, but we might. And then the energy that is released from these electrons transferring from glucose going to oxygen, the energy will be stored to generate ATP. So ATP will store the energy by chemiosmosis, and we'll talk about chemiosmosis later in this lesson. Now, in these redox reactions here, you'll note that NADH is the, oh gosh, what is this thing called? It loses its electron, so it is oxidized, and then, is that right? Loss of electrons. Yeah, it's oxidized, and then FMN gains the electron, so it's reduced, and then the electron goes to the second one, so this becomes oxidized, that becomes reduced, and the point is that down the chain, redox reactions occur, meaning one will be oxidized and the next will be reduced, etc. All right, any question about that? Is this complex? This is just the summary, and the important part here is respiration is one of three ways that cells have for generating ATP. And you note that I'm calling it respiration, not aerobic respiration, because there are other forms of respiration. And we'll talk about aerobic respiration, and then later in this lesson we'll talk about anaerobic respiration. The third way that cells can generate ATP is by photophosphorylation. This happens only in photosynthetic cells where light releases electrons from chlorophyll. The light energy is converted to chemical energy via an electron transport chain, and we'll talk about that later in the lesson. Ultimately, ATP and NADPH are generated, and they're often used to make sugar. So that usually in photophosphorylation, the energy from light is stored as sugar. Okay? And photophosphorylation only happens in photosynthetic organisms, such as shown in this picture here. They're obviously photosynthetic. Why do you think they're so muddy? All right, any questions?
No.
All right. That was a joke about the boys being photosynthetic, by the way. Let's talk about the metabolic pathways of energy production. The energy released from molecules is controlled and stored by a series of reactions. The energy is not released as a single energy burst, meaning when our cells release the energy from glucose, it doesn't happen in one reaction. I could get it to happen in one reaction. I don't know if you can see this, so I'll move it over here. If this cup were to have glucose in it, which actually it has juice in here, which has a little bit of sugar in it, I could release the energy in the glucose in one step by taking a blowtorch to the glucose and then lighting the glucose. And then the energy would be released all at once, releasing a tremendous amount of energy. So much energy that if I were to do that, I would have to stand back from the glucose burning because so much energy would be coming off, it would burn me. Our cells do not release the energy from glucose in a single burst because it would be too much energy at one time for our cells to handle. They control the energy release by a series of redox reactions. The electron from the donor molecule, such as glucose, are passed from one compound to another through a series of redox reactions. And I actually showed you that right here. You don't need to memorize this, but this is the redox reaction controlling the release of the energy that happens starting from glucose. The electrons from glucose flow to NADH and then flow down these electron transfer molecules in the electron transport chain and then end up flowing to oxygen. Any question about that? And that way, the cell can survive the energy release because it's not a temperature that is so hot that it'll kill the cell. Most organisms use as their primary source for cellular energy the molecule of glucose, but they can use carbohydrates. So every time you eat something, you will convert the carbohydrates in your meal into energy. Unless, of course, you eat more than you should, in which case some of it will be converted into fat and you'll store it down that way. But that will be eventually converted, will be burned eventually at one time or another. I mean, you could keep storing more and more fat, but eventually that too would be converted into our energy needs. So many organisms use catabolism, that's my main point here, to get their energy needs from a catabolic molecule such as carbohydrates and glucose. And most of us, when we eat a carbohydrate, it will be converted into glucose and then sent down aerobic respiration as glucose. Any question about that? Most of us, not all of us, if you're a marathon runner, you don't do this. Most of us will convert lipids that we eat, meaning fats in our diet, we will convert those into glucose and then send it through aerobic respiration. A marathon runner, on the other hand, or some Olympic athlete, can directly burn fats. However, they don't do that until 45 minutes into their marathon run. So they have to run for 45 minutes before they start burning fats. And in the 45 minutes before that time, they are burning glucose and other carbohydrates. But we can use lipids and proteins for catabolism to get our energy needs. Most of us who are couch potatoes will convert lipids and proteins into carbohydrates and then convert those into glucose and send them through as aerobic respiration. To produce energy from glucose, microorganisms and, well, actually people only use, yeah, people do too, can use two general processes for getting their energy needs: respiration and fermentation. And our cells can use aerobic respiration, and our muscle cells can use fermentation to get our energy needs. Microorganisms can use either respiration, and some of them use aerobic respiration, others use anaerobic respiration, and some use fermentation. Let me see if I can get this going. I had a hard time getting that going. We might not be able to finish that.
All cells need energy to stay alive. Usually your
Okay, it started with all cells need energy to stay alive. I'm turning up the volume here. I don't know why it's so quiet.
cells get their energy through the process called aerobic respiration. The word aerobic refers to oxygen. In aerobic respiration, oxygen interacts chemically with the molecule of glucose. The glucose is broken down into carbon dioxide and water. This process releases energy, which the cell stores in the chemical bonds of ATP. The first stage of aerobic respiration, glycolysis, occurs in the cytosol, the jelly-like part of the cytoplasm. Glycolysis breaks each molecule of glucose into two molecules of pyruvate, energizes two electron carrier molecules, and produces two molecules of ATP. The next two stages of aerobic respiration are carried out in the mitochondria. Pyruvate enters a mitochondrion. Once inside, it is modified and enters the Krebs cycle. The reactions of the Krebs cycle break the pyruvate molecules down to carbon dioxide, create two more molecules of ATP, and energize more electron carrier molecules. The final stage of aerobic respiration is electron transport. The electron carrier molecules that were energized in the earlier stages deliver electrons and hydrogen ions to the electron transport chain. As the electrons are passed down the chain, energy is released and used to make ATP. This final stage produces as many as 32 molecules of ATP from each molecule of glucose.
Okay, any questions?
No.
All right. We only have 3 minutes left, so I think I'll stop right here. And if there's no questions, I'll see you. Well, I'll be here for the lab, but I'll only be here to 6:30 to 6:45. And if there are no questions at 6:45, I'm going to log off the lab. So if you have any questions, I'll be in the lab from 6:30 to 6:45. Are there any questions for this lecture here?
No, thank you.
Thank
All right, I'll see you whenever. Maybe in the lab, maybe later.